In this video, we present a new visual analytics approach to high-dimensional logistic regression modeling and its application to an environmental health study. The approach consists of 3 interactive views to support a 3-step visual analytics pipeline. The 3 steps are dimension reduction, variable relationships analysis for model building, and model evaluation. This is the univariate analysis view. Users conduct dimension reduction here by selecting variables based on univariate analysis results. Univariate analyses estimate the correlation or association between an explanatory variable and a response variable without other variables considered. In this example, the explanatory variables are maternal exposure to 449 chemicals, and the response variable is whether a birth is involved in limb reduction defects. In this table-like view, the maternal exposures to the chemicals are displayed row by row. The names of the chemicals are displayed in the first column. The columns show the point-by-serial correlation, walled test p-value, crude odds ratio, and odds ratio for low, medium, and high exposure to the chemical. Red indicates risk. For example, you can see here that benzyl chloride has a long red bar in the RPB cell, which means that it is highly correlated with the birth defect. Its walled p-value and odds ratios also indicate significant association with the birth defect, indicated by the red in the cells. The chemicals can be sorted by one or more indicators. For example, clicking the RPB column title will sort the chemicals by their RPB values. Clicking the comprehensive sorting button will sort the chemicals by the maximum of the confidence intervals of their 4 odds ratios. For example, dioxin and dioxin-like compounds is on the top since the whole chemical has a strong association with the birth defect, while cyanazine is on the top since its high exposure group has a strong association with the group. They are all worth to be carefully examined in further analysis. We set both RPB p-value and walled test p-value threshold to 0.25 and filter out with non-significant chemicals. Then we sort the chemicals by the lower bound of their crude OR values. We select significant chemicals by 2 mouse clicks. We click this button to send the selected variables to the variable group view. This is the variable grouping view. It allows users to examine the correlation and confounding among the variables. According to the information, users can interactively select variables to build models. We notice 3 extra variables in the view. They do not have the small yellow indicators in front of their names. They are variables correlated to variables we selected. We click the red label of cyclohexane, variables confounding it are highlighted in green. We click the green label of tetrabromobisphenol A, and variables confounded are also highlighted. We provide a navigation tool for the grass plot. It can be used to explore the stability of the variable and the presence of confounding as other variables included in the model. For example, cyanazine confounds 6 chemicals because there are 6 green blades in the grass. One of them, cyclohexane, leads to an extreme confounding of cyanazine. We switch to the sort mode. We can click a variable to place the cases and controls with high exposure to this variable to the center of all the squares. We sort the data by a variable in the group 0. We find the group 0 and group 11 have strong correlations since their squares have similar textures. We then switch to variable selection mode and start model building. We select independent chemicals with different textures for inclusion in a model and evaluate the model by clicking the button. The results are shown in the model evaluation view. The first column shows the name of the variables. The second column shows the univariate logic model results of the variables. The last column shows the results of the model we just built. We can see that there is no big difference of rectangle lengths between the univariate logic model results and the multivariate logic model results. It means that no confounding is involved in the model. Now we inspect the weak effect using this view. From the variable groups view, we can see that there are three variables having similar textures. They are 1,2-dichloropropane, 1,1,2,2-tetrachloroethane, and 1,1,2-trichloroethane. We add all of them into a model. They all have a gray bar, which means they are non-significant. We only add 1,2-dichloropropane to the same model. The red bar shows it is significant. The same patterns are found for 1,1,2,2-tetrachloroethane and 1,1,2-trichloroethane. So we can say they have a weak effect on the birth defect. We now study the influence of categorical variables on the birth defect. We click maternal age group to add it to the model. A rose plot is displayed on the left. The sector representing mothers above 40 years old is red. It means that it is a most vulnerable group. The results of the modified model are shown in the column after the results of the original model. We can see here that MANEB increases odds ratio from 2.454478 to 2.468568.